So you are expert in genetics. So I just wanted to ask you, how does genetics affects the fertility treatment outcomes to increase the success rate and reducing the uncertainties?
Genetics plays a very important role, especially in context of assisted reproductive techniques, and it complements IVF processes when there are the necessary indications for it. So, for example, if there is a history of familial genetic disorders in a family, which are inherited, then genetics in form of pre-implantation genetic testing for monogenic disorders can help in selecting for embryos which have not inherited those mutations. And the other common use of genetics in context of IVF is pre-implantation genetic testing for aneuploidies, which are chromosomal abnormalities. And it is very well known and very well documented that chromosomal aneuploidies increase with advancing female age. So yeah, pre-implantation genetic testing for aneuploidy, it helps basically in selecting against the aneuploid embryos. And by transferring embryos which do not have the chromosomal aneuploidies, it helps in reducing the miscarriage rates, and it helps in decreasing the time to pregnancy.
Yeah, that's actually very important. In like nowadays, female wants to conceive lately.
Yes.
They want to have the career first and everything, and so there are chances of getting the aneuploidies embryos. So I guess genetic testing plays a very important role.
Yeah, and of course, as I said, that it is quite important when there is particular indications. So the indications like advanced maternal age, which is usually defined after the age of 35. Recurrent implantation failure history or recurrent miscarriage history in patients. These are the patients who would benefit from PGTA. For monogenic disorders, of course, there's already a known history in the family. So these are the patients who can avoid having a child with these inherited conditions if they choose to go for pre-implantation genetic testing.
Yes. So what are some common myths or misconceptions you come across as a genetic specialist in fertility treatment?
So as we were previously discussing, there are a range of genetic tests, pre-implantation genetic testing, which could be performed on embryos. And once patients have undergone these treatments, and if they transfer, let's say, for aneuploidy testing, if they transfer a euploid embryo or an embryo which does not have a chromosomal abnormality, then they think that the pregnancy is guaranteed or live birth is guaranteed. But that is one of the misconceptions because statistically, literature shows that even when you transfer a euploid embryo, on an average, the live birth rate or the success rate is around 70%. So there are still a lot of other factors which could influence the success of IVF treatment, and it's just not dependent on genetics or chromosomal aneuploidies. So, for example, there could be uterine factors, other embryonic factors, there could be mitochondrial factors. So, a lot of underlying factors which we don't really have much clarity on at this stage. And yes, so genetic tests which lead to identifying embryo without an abnormality, even when they're transfers, the success rate is not 100% guaranteed. So that's one of the misconception, or let's say a sort of like a notion which patients sometimes misunderstand. Even if we transfer a chromosomally or a genetically normal embryo as such, it cannot guarantee 100% a live birth from that.
That is true, actually. If we tell patient it is a healthy baby, we are transferring, then they'll feel like now it's healthy, it will get implanted. But they don't know that there are quite a lot of factors apart from only the blastocysts which you are transferring. There are endometrial lining, there are uterine receptivity, everything else.
And the other thing is, which of course patients are consented for when you perform the testing, that if you're looking for chromosomal aneuploidies or a certain chromosomal genetic disorder, even if it results in a pregnancy, there could be a small chance of other abnormalities which the fetus can have, like congenital abnormalities, and in population it usually occurs at approximately 3% of frequency. And these are the things which we are not routinely screening for, or they don't have a genetic basis for, which you could screen for in an embryo. So that general risk which is present at a population level, that still applies, of course, for IVF patients as well.
So looking forward, what advancement in the field of genetics excites you the most, and what do you think that how it will shape the fertility treatment in future?
So at the present moment, we are moving very fast towards the era of personalized genomics or personalized genetics. And that is applying to basically the entire healthcare industry in general or the genetic space in general. And it is something which is already being applied in terms of in terms of the assisted reproduction in form of, let's say, PGT for polygenic risks. And there are certain companies in the US which are offering it. However, even though the space of personalized genetics is very exciting, we need to be cautious about it and ensure that whatever new technologies or whatever new form of genetic screening which we are applying to embryos, first of all, the method has to be properly validated. So we call it analytical validation, meaning whatever you are screening for, that is screened at an accuracy which is medically acceptable. And the second thing is about clinical utility, meaning when you're performing a screening or a test, whether it has clinical benefits and whether those benefits have been proved through means of different clinical trials and studies. So we have seen time and again in the field that many of these tests have been prematurely already offered or prematurely applied in the field. And then later on, studies have shown that many of these add-on tests to IVF, they do not have so much of clinical utility. So we also need to be cautious whenever it comes to new technologies. But of course, the exciting thing right now in this space is moving towards personalized genomics. But it is going to take some time before its clinical validity is shown and whether it is something which is useful at a population scale, that's to be seen yet.
Yeah, because it's personalized, so every person may have their own.
Exactly. So individualized care, but we don't really have, although we are generating genomic data at scale and more data is being available since sequencing is becoming cheaper and more accessible, still the wider outcome data when it comes to things like polygenic risk scores, whether, let's say, if you have diagnosed certain embryos at risk of certain polygenic diseases like diabetes and stuff, that sort of outcome data is long-term, right? You'll only come to know if you do a long-term follow-up study of decades in these babies which are born after these tests. That is missing, simply missing at this stage. So the clinicians and practitioners need to take these sort of tests with a pinch of salt, and they also need to ask for the validity and sort of the clinical and the analytical validation of these sort of tests and the platforms which are used to perform these tests before they're being offered to the patients.
Yeah, that is very true. Now that AI is coming in picture, like AI is everywhere, wherever you see. Do you see that in genetics also there is a scope of AI in future?
Yeah. It's helping you to analyze data at scale and at speed. So with sequencing, you're generating a lot of data and you're generating it at a very fast pace as well. But traditional methods of analysis, it takes quite some time, and especially if it's manual, it's very cumbersome and it's time consuming. And it probably is not going to give you as much information as, let's say, AI tools might give you, because it, of course, can be designed to look at various different aspects of the data set and probably give you something which is meaningful. So that's where it's helping, especially in terms of the genetic data analysis. For example, companies like DeepMind, which is a branch of Google, they came with the AlphaFold predictive model, right, which is looking at the prediction of all the different sorts of proteins which could be formed, and it has really accelerated the biotechnology, like research industry also in terms of drug metabolism and things like that. And recently they came up with another tool which is looking at the DNA and the variants in the DNA and how it might affect the protein functioning, which of course then affect your normal like well-being of the body. So AI tools are really helping in understanding the role of DNA, the role of like other things physiologically in the body and how it may inform the health outcomes. So it is definitely something which is very much part of our, let's say, day-to-day patient care, be it on the embryology side of things, where you can see all of your algorithms, for example, for choosing the right gametes or choosing the right embryos. And on genetic side, it is helping us understand the different variants which are present in the DNA and how that might be helpful in interpretation of those variants and what potential impact it might have in the embryos and of course the future babies. So yeah, it goes hand in hand with our day-to-day work at the moment.
Because I was reading something and I got to know that AI can now find a gene which can something which can responsible for the cancer in future. So it will develop to a cancer. It can find it at a very early stage before the development. So you can just get it.
Yeah, as I said, because we have, if you talk about DNA, right, like it's 3 billion bases, which all of us individuals are from. Every cell has 3 billions and we have trillions of cells. So out of these 3 billion bases, what, of course, geneticists are endeavoring and what traditional genetics looks like in terms of the disorders is you're looking at which variations or which sort of like mistakes in the DNA code lead to what sort of disorders. But we have knowledge about a set amount, like very small fraction of this DNA sequences, which actually is what we call protein coding. But the rest of the DNA, which is not coding for proteins, we didn't really have much information on it in terms of what sort of changes in those sequences can later on impact or has an impact on health. So this is something which the AI is actually helping in doing. So maybe this example which you're quoting is coming from there, where this sort of changes or variations in the DNA, it basically has a downstream impact on some sort of like protein function, and it predisposes towards certain disorders like cancer or predisposition or other health conditions in future.
So maybe in future we can see fertility treatment leading to like the genetics which we do PGTA can give the results via AI, and then will be very too much of precision.
So this is coming back to the era of precision medicine. This is precision medicine because, of course, while DNA is different between individuals, most of the genes which are well characterized, and we know that mistakes in the code of these genes cause these disorders. That is what we call Mendelian inheritance and Mendelian disorders. But then there are other parts of the DNA where changes or what we call polymorphisms are specific to certain ethnicities and certain populations, and they have different impact on different ethnicities and populations. And these changes together, we call them polygenic because they are in multiple genes. And the polygenic changes can have basically different impacts. So in different ethnicities, for example, one ethnicity which has a risk predisposition towards diabetes or other health conditions like cardiovascular conditions and things, these are things which can be impacted by changes in DNA, and then of course environmental and lifestyle changes as well. So that's where polygenic risk scores comes in place, where there are now these tests which some groups are offering to screen for these sort of like polygenic alterations in the DNA of embryos. So during PGT, they will test for this, and then they will rank the embryos. And they say, okay, this embryo probably has a lesser chance of developing diabetes in future, so transfer this. So that's where I was saying to be cautious, because even though there is science behind it, and there are resources where you know what sort of polygenic risk scores inform what sort of decisions in terms of embryo selection, we don't have the outcome data. So it's not proven yet that in embryos that we have enough knowledge that the way in which we are ranking the embryos, that long term it is going to cause that impact, or if it's going to avoid the disorder. So we need to be cautious about those. But we are in the era of precision medicine.